Assalamu alaikum. My name is Hajra Yunus, and in this lecture and today's lecture we will discuss about uh, eukaryotic end replication problem and one more topic, that is mitochondrial DNA replication. So how in mitochondria DNA is replicated. So in today's lecture we will cover these two topics. Now in this slide we will discuss about end replication problem in case of eukaryotes. As you know that in prokaryotes there is no end replication problem. So why prokaryotes do not have end replication problem? So answer is pretty simple, and it is because of the circular nature of DNA. As you know that the prokaryotes of the bacteria have the circular DNA. Because of circular DNA, here the 5' phosphate group is available and here the 3' hydroxyl group is available. And every time there is availability of 3' hydroxyl group to DNA polymerase, that's why in case of prokaryote there is no end replication problem. And replication problem is faced in the eukaryotes because of the linear DNA. As you can see in this diagram, here the DNA is linear in nature, and because of the linear in nature there is a end replication problem. So let me explain this diagram to you. What is end replication problem? As you can see in this diagram, this strand is the template strand, and opposite to that complementary strand is formed. So here the primer is, this is one primer, and here the another primer is present. And as you know that in case of eukaryotes, the primer is formed by polymerase alpha, and polymerase alpha has two subunit, and the smaller subunit is responsible for formation of primer. So after the completion of DNA replication, the primers are removed, and after the removal of primer, as you can see here, the space is available. So here the gap is present, and these gaps are filled by polymerase epsilon and or polymerase delta. But as you know that here the 5' phosphate group was available, and here 3' hydroxyl group is available. And because of the availability of the 3' hydroxyl group, DNA polymerase can perform the function of polymerization. And here the, as you can see here, the gap is filled, but here the gap is still present. So why the gap is still present here? And the gap is present here because of the presence of 5' phosphate group. As long as there is a 5' phosphate group, DNA polymerase cannot perform the process of polymerization. So for the process of polymerization by DNA polymerase epsilon or DNA polymerase delta, 3' hydroxyl group is available, but there is no 3' hydroxyl group. So you can see here the gap is present. So this gap is not filled by DNA polymerase right now. So this is the end replication problem. Okay, and we are talking about this if there is a gap during the first replication and there is no solution for that. In the second replication cycle of replication, the gap will be present again. This, as you can see, that this much information is not present, and this much information is not transferred to the next cell, or so during the second cycle of replication. So here again there will be the another end replication problem. So this information will not be transferred to another cell, and during the third cycle and so on, this amount of information is not transferred to another cell. So you can see that I have written here that after 40 to 45 times, or after 40 to 45 cycles, the same happens, leading to DNA damage, because the time will come when the DNA has the very important function, because here ending of the DNA has the repetitive sequences. Here the repetitive sequences are present. So if there are the repetitive sequences, so and then there will be no problem, because the information is not very important information. Although the information is not very important, but if there is a gap during the first cycle of replication, and this gap is not filled during the second cycle of replication, there will be another gap, and during the third cycle of replication there will be another gap, and the time will come when the DNA has a very important information, and that information is not transferred to the next cell, so it will lead to the DNA damage. So mostly it is said that after 40 to 45 times, if the same happens, it will lead to the DNA damage. Now there is another question, that in our body a number of cells is present, like somatic cells are present, stem cells, germ cells are present. So why the question is that these somatic cells are more affected by the end replication problem compared to the stem cells or the germ cells. So answer is pretty simple. It depends on the production of telomerase, because telomerase will help in resolving the end replication problem. So telomerase enzyme is produced by stem cells and the germ cells, but the somatic cells cannot produce or do not produce telomerase enzyme. So now there is another question: why somatic cells do not produce telomerase enzyme, but the stem cells and the germ cells can produce the telomerase enzyme? So answer is pretty simple, and that is, it depends on the expression. Although the gene for the production of the telomerase is present in somatic cells and the stem cells and germ cells also, but the thing is that it depends on the expression. In the stem cells and the germ cells, there is expression of telomerase, but in case of somatic cells, there is no expression of telomerase. So that's why somatic cells are affected more by the end replication problem compared to the stem cells, germ cells. And the cancerous cells, there is one more thing: cancerous cells have also the ability to produce telomerase. Okay, so if there is a question uh, which cells do not have the ability to produce telomerase, so answer is somatic cells. And if there is a question that which cells have the ability to produce telomerase, so answer is stem cells, germ cells, and cancerous cells. Okay, now in this slide we will discuss about how telomerase is responsible to end the end replication problem. So as you can see in this diagram here, there is a end is unreplicated. Okay, as you can see, why end is unreplicated? Because here 5 prime phosphate group is present, and because of the 5 prime phosphate group, there is a missing DNA on the lagging strand. Okay, we will discuss with respect to the lagging strand, or we will discuss in context of lagging strand, but in case of the leading strand, the procedure is similar. So whether we talk about lagging strand or leading strand, in both cases the procedure is similar. So how, the next question is how telomerase would help in, or telomerase helps in solving the end replication problem. So because the telomerase has its own RNA template, and because of the presence of its own RNA template, so the complementary base pairs are added. So as you can see here, the blue color is the telomerase, and the telomerase has its own RNA template, and which is shown in yellow color. And here opposite to that, or complementary to that, DNTPs are added. And now as you can see here, now because of the attachment of the RNA template, and here the 3 prime hydroxyl group is free. Now because of the availability of 3 prime hydroxyl group, now the DNA polymerase will perform the process of polymerization and will complete the process of replication. But there is one more thing you need to know, that there is the telomerase extends unreplicated end. What is meant by telomerase extends unreplicated end? So it is meant by the telomerase continuously or telomerase keeps moving forward. So as you can see that here the telomerase firstly in this diagram was here, and after that the telomerase was here. So continuously telomerase is moving, and continuously telomerase is responsible for adding new or for adding new DNTPs. So as you can see that here the length of the DNA was here till now, and in the next step the DNA length of the DNA is till now. So in case of the telomerase replication or end replication problem resolve, or how the telomerase is responsible to resolve the end replication problem. Firstly, the telomerase has its own RNA template after attaching to the DNA strand. So now the RNA template is present, and opposite to that DNTPs are added, and after the adding the DNTPs, telomerase will move forward, or telomerase keeps moving forward, and because of the uh, uh, forward movement of the telomerase, now again DNTPs are added. But there is one more thing: 3 prime OH group is available, and because of the availability of 3 prime OH group, now DNA polymerase can perform its function and will complete the process of polymerization. Now in this slide we will discuss about how long telomerase will move forward and would be responsible for the addition of more DNTPs. So as you can see in this diagram, two things are very important. Number one is the activator, and the activator is the CDC13, and there are different proteins are also present, that is RIF, RAP2 and RAP1, which are also called RIF-RAP proteins. So these things are very important. So the question is that so let me tell you the functioning of them. So the CDC13 is responsible to let the telomerase attach on the DNA strand. Okay, so and CDC13 has more affinity for telomerase, whereas in addition to that, as you know that there are other proteins are also present, that is RIF1, RIF2, RAP, which are also called RIF-RAP proteins, and they have less affinity for telomerase. So why the RIF-RAP proteins have less affinity for telomerase compared to the CDC13? So the answer is that because of their number is less, and the function of RIF-RAP proteins are to inhibit the uh, activity of telomerase enzyme. So right now at this step, CDC13 has more affinity for the telomerase. That's why the telomerase will attach to the DNA strand and will move forward and will be responsible for adding more DNTPs or new DNTPs. But on the other hand, RIF-RAP proteins have less affinity compared to the CDC13 because of their less number. As you can see here, 1, 2, 3 and 4. Here the four RIF-RAP proteins are present, and because of less number of RIF-RAP proteins, their affinity is less, and because of their less affinity, so they would not be able to inhibit the activity of telomerase enzyme. So now, as you know that here, now in the next step, the telomerase has attached, and in the step 3 you can see that telomerase is moving forward and telomerase keeps moving forward. And here, as you can see that here DNA polymerase has performed its function. Telomerase, as you know that in case of eukaryotes, telomerase alpha or telomerase epsilon is responsible for the polymerization of the DNA strand. Like for the case of leading strand, the DNA polymerase epsilon, and for lagging strand DNA polymerase delta is responsible for uh, process of polymerization. As you can see that here, now the double stranded or the double strand has formed, and now as you know that the more RIF-RAP proteins are being attached or firstly, and here in the last step you can see that here the number of RIF-RAP proteins are more. So firstly, there were in the first step or in the beginning there were only 4 RIF-RAP proteins, and in the end now you can see here the 1, 2, 3, 4, 5, 6, 7 and 8 proteins are present. And now at that time or right now, because of the large number of RIF-RAP proteins, now oh, they would be able to inhibit the activity of the telomerase enzyme, and they will force the telomerase enzyme to leave the DNA strand. Firstly, in the start or in the beginning, CDC13 has high affinity because of the less number of RIF-RAP proteins, and now after the attachment of the telomerase enzyme, telomerase uh, has the RNA template, and opposite to the RNA template or complementary to the RNA template, DNTPs are added. And after adding DNTPs, now 3' OH group was available, and because of the availability of 3' OH group, now DNA polymerase was able to add more DNTPs and will continue the process of polymerization. After completing the or after forming the double strand structure, now here the double strand was available, and RIF-RAP proteins can only add on the double strand. Now in the end you can know that more RIF-RAP proteins are available, and now they will force or they will stimulate the telomerase to leave the DNA strand, and that's how the telomerase enzyme leave the DNA strand, and there is no more further or there is no further elongation of DNA strand. Now as you can see in this diagram, after removing the or after leaving the telomerase, as you can see here the telomerase has left the DNA strand, but here the length of the DNA strand has extended or has increased. So now Next step is the folding of the DNA strand. So why the folding is required? Folding is required to protect the DNA strand from the exonuclease activity, because if there is a exonuclease activity, there will be the DNA damage in the end. So protect the DNA from the exonuclease activity or from the DNA damage, folding is very important. As you know that here in this step, the folding means the DNA is inside. And why the DNA is inside? The DNA is inside to protect the DNA strand from the DNA damage. And in the next step, here is the DNA invasion. In that case, the T loop is formed. As you can see here in this region, you can see the T loop. Okay, so this is basically how the end replication problem is solved in case of the eukaryotes. So basically, if I summarize the whole end replication procedure, so it is pretty simple. Like in case of prokaryotes, there is no end replication problem, but eukaryotes have end replication problem because of the linear structure of DNA. So because of the linear structure of DNA, in the end 5' phosphate group is present, and because of the availability of 5' phosphate group, DNA polymerase cannot perform the process of polymerization. So in that case, telomerase, which is produced by DNA germ cells and the stem cells, and in case of the cancerous cells, they have the ability to produce the telomerase enzyme. So because of the end replication problem, telomerase comes and attaches to the DNA strand. So why telomerase? And the question is that how telomerase would help in resolving the end replication problem? Because of the RNA template, because the telomerase enzyme has the RNA template, and in complementary to the RNA template, DNTPs are added. And now because of the presence of or attachment of the RNA template, 3' hydroxyl group is available, and now DNA polymerase can proceed further or can complete the process of DNA polymerization. But there is one more thing, that the telomerase enzyme is continuously moving forward, or DNA polymerase telomerase enzyme keeps moving forward. And now the another question is that how long telomerase enzyme keeps moving forward. So answer is that, and who would be responsible to inhibit or the activity of the telomerase enzyme? So as you know that on the DNA strand, CDC13 protein is available, and CDC13 protein has the more affinity for the telomerase enzyme. And on the other hand, other proteins are also available, like RIF1, RIF2 and RAP proteins are available, and these RIF-RAP proteins uh, uh, are responsible to inhibit the activity of telomerase enzyme. But firstly, in the beginning, because CDC13 has more affinity for the telomerase enzyme, so telomerase comes and attaches to the DNA strand and would help in resolving the end replication problem. But at that time, RIF-RAP protein has less number. So if the number of the RIF-RAP protein is less, in that case they would not have much affinity as the CDC13 has. So now, in order to get the more affinity, their number would be more. So and how their number would be more? Because they will attach on the double stranded structures. Telomerase can attach on the single strand structure, but for the RIF-RAP proteins, double strand is required. So after completing the process of polymerization, now the double strand is present. Now there will be more attachment of RIF-RAP proteins on the DNA strand. In the end, there are more RIF-RAP proteins. Now the RIF-RAP proteins are more in number, so they will inhibit the activity of the telomerase enzyme, and telomerase enzyme will leave the DNA strand. After leaving the DNA strand, there is a extended end is present, so protect the extending end from the exonuclease activity. There is a folding of the DNA strand, and next step is the strand invasion. In that case, T loop is formed. Okay, so this is basically the whole structure or whole procedure about to resolve the end application problem in case of eukaryotes. Now in the next slide, we are going to discuss about the mitochondrial DNA replication. As you can see in this diagram, this is the mitochondrial DNA, and in case of the mitochondrial DNA, 37 genes are present, and the length of the DNA is, or the base pairs are 16,569 base pairs are present. Okay, and if we quickly look over that, there are different genes are available, and some important genes are rRNA gene is available, tRNA gene is available, ATP synthetase like F0 F1 particle genes are available, which are responsible for the synthesis of ATP, and non-coding genes are also available, complex 1, 3 and 4 genes are also available. So these are very important genes. Now as you know that the DNA is, in case of the mitochondrial DNA, it's a double stranded circular structure. And if the double stranded circular DNA is present, so as you can see here, the outer strand is present and inner strand is present. So this one is the outer strand and this one is the inner strand. Okay, so outer strand is heavy, so I will represent outer strand with the H because the outer strand is heavy. And why outer strand is heavy? Outer strand is heavy because of the presence of purine. So outer strand is purine rich, and you know that the purines are A and G, that is adenine and guanine. Whereas the inner strand, I will represent inner strand with L because inner strand is light. Why inner strand is light? Because of the presence of pyrimidine, and because the inner strand is light, and light strand is pyrimidine rich, and you know that the pyrimidines are thiamine and cytosine. Another important thing is that unidirectional. As you know that in case of eukaryotes and prokaryotes, replication is bidirectional, but in case of mitochondria, replication is unidirectional. Okay, another thing is that, as you know that in case of prokaryotes, there was only one origin of replication. Okay, and in case of uh, eukaryotes, there are a number of origin of replications are present. But as you know that in case of mitochondria, outer strand is heavy and inner strand is light. So in case of outer strand, that is a heavy strand, another or separate origin of replication is present, whereas in case of the inner strand, which is light, and for that the separate origin of replication is present. So as you can see here, the OH, this is the OH, and OH is the origin of heavy strand synthesis. And here somewhere here, okay, so here the OL is present, and this OL stands for origin of light strand synthesis. Okay, so let me revise this slide one more time. That is this is the mitochondrial DNA, and the mitochondrial DNA has 37 genes, and the length of the or number of base pairs in the mitochondrial DNA is 16,569. Okay, so some important genes in case of the mitochondrial DNA are RNA, tRNA, ATP synthesis, non-coding, complex 1, complex 3, and complex 4 genes are available, and many other genes are available. As you can see here, so many genes are available, but these genes are very important. Okay, now the mitochondrial DNA is circular in nature and is double circular structure, not the helical structure. It's the double stranded circular DNA. Now it's the double strand circular DNA, so outer strand is different and inner strand is different. Okay, in case of prokaryotes and eukaryotes, there is no differentiation and there is no dissimilarity between the outer strand and inner strand. But in case of mitochondrial DNA, the outer strand is heavy and inner strand is light. Why outer strand is heavy, inner strand is light? Because in outer strand, outer strand is purine rich and inner strand is pyrimidine rich. And another thing is that in case of mitochondrial DNA, replication is unidirectional, and for outer strand there is separate origin of replication is present, and for inner strand there is separate origin of replication is present in case of mitochondria. But as you know that in case of prokaryotes and eukaryotes, there is a single origin of replication is for the double strand. But in case of mitochondria, for the outer strand, OH is the origin of heavy strand replication and OL is for the origin of light strand replication. Now in this slide we will discuss about how the mitochondrial DNA replication is proceeded or is completed. Now as you can see in this diagram, so this is the heavy strand, and the heavy strand is represented by black in color, and the inner one is the light strand, and the light strand is represented by light gray color. And here they For the, as you know that for the heavy strand the original replication is different, so OH is the original replication for heavy strand, and for light strand original replication is different, so here the OL is the origin of light strand synthesis. So here firstly the replication will start on the heavy, will start from the heavy strand. So as you can see here in this diagram, here the nick is created. Okay, as you remember that we discussed about the prokaryotic replication and the eukaryotic replication, and none of the replication the nick was created, but in case of the mitochondrial DNA replication the nick is created. Firstly the nick is created, and after creating nick the heavy strand, so this one is the heavy strand, this heavy strand is pulled outside, and after pulling outside single stranding binding proteins attach on the DNA strand, and after, you know that what is the function of the single strand binding protein. Single stranded binding proteins are responsible to prevent the rewinding of DNA strand. Okay, so here the green ones are the single stranding binding proteins. And there is one more thing you need to know, that in case of the mitochondrial DNA the polymerase is also will perform the function, helicase is also involved, and the primase is also involved, but all these things have different name. Like you can see here the polymerase gamma is the DNA polymerase that will help in the polymerization of DNA strand. Twinkle is the helicase which will help in the unwinding of DNA strand. Pol RMT is the primase which is responsible for primer formation. Okay, so as you can see, firstly the nick is created, and after creating nick the heavy strand is pulled outside, and single stranding binding proteins will attach to the DNA strand to prevent the unwinding of rewinding of DNA strand. Now here the firstly by using the light strand heavy strand will form. Okay, this is very important. Okay, firstly strand heavy strand synthesis is formed. So if the light strand is used, in that case heavy strand is obtained, and you know that if the heavy strand is used, in that case the light strand is obtained. But firstly the heavy strand is formed. So here come the this is the light strand, this light strand is used as a template, and here the pole RMT is responsible for the primer formation. Firstly primer is formed, and here polymerase gamma is responsible for the synthesis of the heavy strand. As you know that here the primer is available, and the polymerase gamma is performing its function, or is performing the process of polymerization. And you know that we discussed in the prokaryotes that the helicase and the DNA polymerase, they both are associated. In that case their activity is improved, but if they dissociate from each other, in that case their activity will affect, and helicase will move faster, and DNA polymerase, and in that case when the DNA polymerase is proceeding the process of polymerization, in that case it will encounter the tangled DNA. So in that case that's why they are associated to each other for the efficient DNA replication. And now you can see that here in the third step that here the loop is present. Okay, so you know that after 2 to third completion of heavy strand, then replication starts on light strand. So this is very important. Okay, you know that by using the light strand heavy strand is formed, and after completing 2/3 synthesis of heavy strand, then replication starts on the, or starts for the light strand. So here you can see here the loop is present, and because of the presence of here, because of this loop here the helicase, and now because of the presence of loop here single stranding binding proteins are not attached to the DNA strand, and now here the primer will attach. You can see that here the single stranding binding proteins are not available, there are no single stranding binding proteins. So here the pole are empty, that it is a primase, then the primase will help in the formation of the primer, will form the primer, and DNA or DNA polymerase, that is the polymerase gamma, will synthesize the light strand by using the heavy strand. So as you can see here there is a simultaneous synthesis of light strand and heavy strand, or in the end you will get the heavy strand and you will get the light strand. So the procedure is pretty simple. The first step is the nick formation. The nick is formed, and after creating nick, the heavy strand is pulled outside, and on that the single stranding binding proteins are attached to that. What is the function of the single stranding binding proteins? Single stranding binding proteins are responsible to prevent the rewinding of DNA strand. Now, firstly, in the first step, by using the light strand, heavy strand is synthesized, and firstly POLRMT works as a polymerase primase that will form the primer, and then polymerase gamma is responsible for the process of polymerization. After completing 2/3 of the heavy strand synthesis, then the replication starts for light strand synthesis. Okay, so by using the light strand, heavy strand is formed, and by using heavy strand, light strand is formed. And after 2/3 completion of heavy strand, as you can see that here the loop is present, and because of the loop formation, here there will be no attachment of single stranding binding proteins. In that case, the primer is responsible, primase is responsible, and that is the pol RMT for the primer formation, and the DNA polymerase, that is the polymerase gamma, will perform the process of DNA polymerization, and there is a simultaneously replication of the light strand and heavy strand, and in the end you will get the light strand and heavy strand. You will get the uh, replica or two copies of the DNA, mitochondrial DNA. So this is the whole procedure how the DNA, mitochondrial DNA, is replicated. So today's lecture we discussed about the end replication problem in eukaryotes, and you know that in prokaryotes there is no end replication problem, and we discussed about how the end replication problem is resolved, and we discussed about how telomerase will help in resolving the end replication problem, and we also discussed about after resolving the end replication problem when and how the telomerase would leave the DNA strand. And you know that for that the refrap proteins are important, and the number of the refrap proteins is very important. And now after that we discussed about the mitochondrial DNA replication. We discussed, or we looked at the structure of the mitochondrial DNA, like how many genes are available and how many base pairs are available, and we also uh, looked up on the important genes in mitochondrial DNA, and we also discussed about that in case of the mitochondria, the DNA replication is unidirectional, and the light strand and heavy strand, two different strands are present. And now because of the two different strands, here the original replication for the light strand is different and the heavy strand is different. And now in the last slide we discussed about that in case of mitochondrial DNA replication, firstly nick is created, and you know that in case of prokaryotes and eukaryotes there is no nick formation, and then the heavy strand is stretched out, and the single stranding binding proteins attach to the DNA strand, and after attaching to the DNA strand, here the by using the light strand, heavy strand is formed, and pole RMT is used as a primase which is responsible for primer formation, and polymerase gamma is responsible for the process of polymerization. After 2/3 completion of heavy strand, now the synthesis or on synthesis, that application starts for light strand by using the heavy strand. And here the loop is present because this loop, here the double stranded structure is available, and as long as there is a double stranded structure, single stranding binding proteins will not attach to the DNA strand, and now there is a simultaneously formation of single strand and light strand, heavy strand and light strand, and in the end two copies of the mitochondrial DNA replicates are obtained. So this is the whole procedure. I hope you get or you understand the whole procedure of mitochondrial DNA replication and how to resolve end replication problem is resolved by telomerase enzyme. And if you have any question, you can ask you can ask me writing your question in the comment section, and do subscribe this channel if you have not subscribed this channel, and also click the bell icon button. In the future if I upload any video, you will get the video. Till then, Allah Hafiz.